It's my great pleasure to invite Dr. Dimitris Panagopoulos from Athens to come and speak to us next. Dr. Panagopoulos has a degree in physics and a PhD in biology from the University of Athens, and he's an EMF-biophysicist who works at the Laboratory of Health Physics, Radiobiology and Cytogenetics in the National Center for Scientific Research, the Demokritos in Greece. His experiments were among the first to show the damaging effects of microwaves and other types of man-made EMFs on DNA and reproduction. And his theory on the mechanism of the action of EMFs on cells is still considered one of the most valid ideas. So without further ado, I'm going to ask Dimitris to come up and talk to us about why modern telecommunication microwave EMFs are more damaging than other types of man-made EMFs. What is the mechanism of action? Thank you very much, Dimitris.
I must thank the organizers for inviting me here, and thank you all for coming. This is the electromagnetic spectrum. This is the ionizing part, the visible part, the infrared, and the part which concerns us is the man-made part. Of course, there are natural fields in this band also. We are mostly concerned about mobile telephony or microwave telecommunications, electromagnetic fields. I will use equally MT for both mobile telephony or microwave telecommunications, because all microwave telecommunications, electromagnetic fields, they all combine an RF carrier, a radio frequency carrier, high frequency carrier, which is pulsed and modulated on ELF, on extremely low frequencies, in order to carry the information, because the information is included on the ELFs, in the ELFs, and in order to increase the density of the transmitted information, they have to pulse the radiation. So we have a combination of RFs and ELFs. We live within a natural electromagnetic environment since the beginning of biological evolution, and every form of life is basically electromagnetic. All cellular functions are controlled by weak endogenous electric currents within the cells and tissues, which consist of ion flows through the cytoplasm and the cell plasma membranes. If we distort these currents, we distort the corresponding biological cellular functions which are controlled by these currents. Natural electromagnetic fields, they interact constantly with the physiological endogenous electromagnetic fields. We are attuned, our brain oscillations are attuned by the Schumann oscillations, by the atmospheric oscillations. The basic frequency of the Schumann oscillations, 7.8 Hertz, is exactly the basic frequency of the alpha rhythms of our brain oscillations. This is not a coincidence. Thereby, it is inevitable that the man-made fields will also interact with these endogenous fields inside cells and tissues and will distort the corresponding biological functions. A very important parameter when we're talking about man-made fields, and a very important difference from the natural fields, is that man-made fields are totally polarized. Natural fields are never totally polarized. There are some of them, like the terrestrial fields and the cell membrane fields, which are locally polarized, significantly, but they are static. Most of the natural fields are unpolarized and oscillating. Anyway, the first picture is an unpolarized field, which is defined polarization by the vector of the electric field. The electric field oscillates on every possible plane. For example, natural light consists of photons. Each photon has a different polarization. One photon oscillates like this, another like this, like this, like this, like this. If we sum all this, if we average all this, the average is zero. So there's no electric field. But on the man-made fields, they are all polarized like this, meaning that the electric field and the magnetic field in another plane, they oscillate on a single and the same plane all the time. So we do have electric field. And they are capable, the polarized fields are capable because of this, to induce forced oscillations on every charged particle within living organisms and everywhere, on every charged particle. In our case, we are concerned about the free ions within cells and tissues, which control practically every cellular and biological function. Only polarized waves of identical polarization and frequency can produce standing interference. This is when we have areas of increased intensity and areas of decreased intensity. This phenomenon was found first by the great English physicist Thomas Young back in the 1800s. Thomas Young found this phenomenon by natural light. Natural light is not polarized. How come it gives interference? Well, my opinion is that this is a dark point for quantum physics. Quantum physics considers that the particle, that the light, that the photons are particles of light, and they can change into waves. So it's black magic. The same thing is both particles and waves. I don't believe in black magic, and I believe I have published this a few years ago that they are not particles. Photons are wave packets, and the reason why Thomas Young produced standing interference by natural light, which is unpolarized, is that he passed the light from a single source from two identical sources. So each photon, each moment passes simultaneously a part of the photon through the one hole and a part of the photon through the other slit. So we have two identical sources of light with exactly the same polarization and frequency. That's why we have standing interference with natural light. There is a great similarity between two natural electromagnetic fields. There's a very intriguing similarity we got to observe and get out very important conclusions. We have the terrestrial electric field and the cell membrane electric field. They are both locally polarized and static. I'm not saying they are totally polarized because, as you see, the vector goes on a sphere, is not parallel the vectors. So, but locally, it is, we can say it's significantly polarized. They are both locally polarized and they're both static. When they don't change, they don't produce effects. When the terrestrial fields change by 20% of their normal values every 11 years, when we have during magnetic storms, due to changes in solar radiation, then we have health effects in human and animal populations. Also, in the cell membranes, we have the voltage-gated channels, which open or close due to changes in the membrane voltage. And they can be gated, open or closed, when a change of 30% in the normal membrane potential takes place. So we have a 20% change in the terrestrial field that produces health effect and a 30% change in the membrane potential that induces cellular effects. It's an astonishing, intriguing similarity. So an important clue here we have that biological and health effects initiate when polarized fields undergo variations on the order of 20 by 30% of their normal intensities. I think we have a lot to learn from this observation. And now we have all man-made fields which are totally polarized. And they undergo all the time, every moment, especially the MT, the mobile telecommunication fields, they undergo changes exceeding 100% of their average values. So we got here an important clue about the reason of the biological effects. The most important sources for electromagnetic pollution are the high voltage power lines, and the MT electromagnetic fields, mobile telephony and microwave telecommunication, including Wi-Fi, DECTs, domestic cordless phones, and all these forms of radiation. They all combine, as I already told you, an RF carrier with ELF pulsing and modulation. I already told you that ELF modulation is necessary for the transmission of information, and ELF pulsing is necessary for increasing the density of information. There is strong experimental evidence that the biological effects of the mobile telephony fields are mainly due to the included ELFs and not to the carrier itself. There have been many experiments. These ones are very good examples of experiments that have compared an unmodulated and unpulsed continuous carrier wave of high frequency with the same wave pulsed and modulated by ELFs, and it was effective biologically only the second case. The RF carrier alone was not effective biologically. This is a carrier wave, and this is the low frequency wave, the ELF, that carries the information. This is amplitude modulation of the carrier by the ELF, and this is frequency modulation. And this is pulse modulation. We have rectangular pulses here, and within each pulse we have the carrier. And then it's zero, and then the next pulse comes, etc., etc. This is how a simulated mobile telephony signal is. This is an ideal figure for a pulsed signal, invariable pulsed signal. This is totally predictable, as you can see. We have the carrier, which can be, for example, in the frequency of mobile telephony, 900 megahertz or 1800 megahertz or whatever. And we can have the same pulse repetition frequency, for example, in 2G, 217 hertz, or in 3G, 100 hertz. And this is simulated signals with invariable parameters. How is a real signal from a mobile phone? It's like this. And how is a real signal from a mobile phone base antenna, from a mobile telephony base antenna? It's much more complicated because at the same time communicates signals from thousands of users, communicates with thousands of mobile phones. So it's like this. So there's no similarity between this and this or this. No similarity. And I believe, I've published this several times. It's not only I believe, there are experimental data showing this. That it is the unexpected variation of the signal that makes it extremely bioactive. Because the living organisms have incredible defense mechanisms, wisely made, and they can adapt to a stressor when the stressor is kept constant. But when the stressor is changing unpredictably each moment, of course, nobody can adapt to it. So we got another important clue that except for polarization, intensity, and ELFs, the unpredictable variability is an important bioactive factor. And this is shown by experimental facts. 50% of the studies that have used simulated signals, as IARC suggests, they suggest that experiments must be performed with simulated signals because we can measure them. It's as if we are seeing a huge tidal wave coming to the shore, and we say, can we measure it? No, we cannot measure it exactly. Oh, ignore it. It doesn't matter. If we can't measure it, it doesn't exist. So if we can't measure it, we don't use it in our experiments, and we use simulated signals, which are so much different, but we can measure them exactly. And so the result is that 50% of the studies, they don't find effects. We are very much indebted to the the personnel of the two big studies. I believe that their biologists did an excellent job, a very accurate job, and they did found effects with simulated signals. Overall, about 60% of the studies, regardless whether they use simulated or real signals, they do find effects. That's a fact. But if we only if we only look to the studies that they used real signals, then 90–80% they find effects. A few words about the model, about the biological model we used in our experiments in the University of Athens with fruit flies. Here's a vial with the food of the flies and a cotton plug. This is how we exposed to mobile phone. And we had constructed coils for magnetic field exposure and a generator for pulsed electric field. So this a few words about the model, about the biological model. These are the ovaries of a female insect. Each ovary consists of about 20 ovarioles. Each ovariole, it's an egg assembly line with egg chambers budding off the germarium, which is the first follicle here, and developing through successive stages until the mature egg is ready to be fertilized and laid. We only studied early and mid-oogenesis because during late oogenesis there is cell death anyway, and we didn't want to study physiological cell death. We wanted to study induced cell death by the fields. So we only studied the eggs from stage 1 up to stage 10, where we do not have physiological cell death and DNA fragmentation. Therefore, studying these stages, if we see DNA fragmentation, we know this is induced, is not physiological. And we used an assay, is a very well-known assay in biology called tunnel assay. In a few words, there's a fluorescent substance called fluorescein, which sticks wherever it finds a broken DNA chain. And when this substance, this fluorescent substance, is incorporated on broken DNA, then when we illuminate the samples with ultraviolet radiation, then this substance emits a characteristic visible radiation, which denotes that it is incorporated on broken DNA. This is the background fluorescence. This is the normal fluorescence of an unexposed sample. This is how physiologically egg chambers should look. And this is an egg chamber, this is an ovariole from an exposed female insect, where we have this characteristic fluorescence denoting DNA fragmentation only in the germarium. Germarium is called also early oogenesis checkpoint, is the most sensitive developmental stage of early oogenesis. When defects are detected, When defect egg chambers are detected by the system itself, the organism, sends them to cell death in order to prevent a problematic offspring. So here we have DNA fragmentation only in the most important, in the most sensitive stage, and the other stages are unaffected. Here we have DNA fragmentation in the second checkpoint, in the mid-oogenesis checkpoint, which are stages 7 and 8, and the other stages are unaffected. This or this were the pictures of the DNA fragmentation with the other fields, with the magnetic field exposure and with the pulsed electric field. We only saw DNA fragmentation in either one of the two checkpoints, of the two most sensitive developmental stages, and not on the other stages. And also, we saw DNA fragmentation in the nurse cells, these cells here with a big nuclei, and in the follicle cells surrounding, in some of the egg chambers, we saw there are follicle cells surrounding the egg chamber which build the egg cell. With mobile telephony fields, with mobile telephone exposure, we saw frequently DNA fragmentation in both checkpoints. Here we see that both checkpoints have DNA fragmentation. And amazingly, in many pictures, in many samples, we saw DNA fragmentation only with mobile telephony fields in all developmental stages, and in all three types of egg chamber cells. The oocyte, which will give us the offspring. You see the oocyte with a small nucleus here? This is a single cell. And the nurse cells which will become nutrients for the oocyte later, and in the follicle cells, can you see the follicle cells with a small nuclei surrounding the egg chamber and building the egg cell? So with previous stress agents, they had not seen cell death or DNA fragmentation in the oocyte, never. With starvation, with dehydration, with chemicals, they never saw DNA fragmentation in the oocyte, and they never saw DNA fragmentation in all developmental stages, but only in either one of the two checkpoints. That was the first time that we saw DNA fragmentation with mobile phone field in all developmental stages and in all three types of chamber cells. So with this model biological system, we compared the effects under identical conditions and experimental procedures with 6 different electromagnetic fields, 3 different magnetic fields with three different intensities, 1, 11, and 21 Gauss. I must tell you that the strongest magnetic field under the highest voltage power line is less than 1 Gauss. And we used 1, 11, and 21 Gauss. One pulsed electric field, I told you before, which resembles the atmospheric discharges from lightning. It has a carrier here and a pulse repetition frequency, 44 Hertz. Where is it? Yeah. And we used 2 different mobile phone fields, GSM 900 and GSM 1800. And we exposed under identical conditions and procedures, as I told you, but with different exposure times. In the magnetic field, it was constantly for 5 days, 120 hours. With the pulsed electric field, was 30 hours in 5 days. And with the mobile phone fields, was 6 minutes per day for 6 days, 36 minutes. So huge differences in exposure duration. And here is the results. Here's the result. This is the DNA fragmentation induced by the two mobile phone fields. 900 megahertz was induced a little stronger field than 1800 megahertz. Here are the three different intensities of the magnetic field. Very similar to those of power lines, but much stronger. And this is the pulsed electric field. There were some previous comparison studies between mobile telephony fields and power line magnetic field, which are very important fields in our lives. These two comparison studies, they use simulated fields, not real fields, and they found comparable effects between the two fields, between the mobile phone field, simulated field, and between the magnetic field. As you saw, in our experiments with the real mobile phone, we found much more stronger effect with the mobile phone than with the magnetic field, even in much stronger intensities than those of power lines. So, there's an important question here. I want you to follow my reasoning, because we can have very useful conclusions from this reasoning, I think. Which specific parameter of the real mobile telephony field is mainly responsible for this intense bioactivity? We know from the whole electromagnetic field bioeffects literature for decades that the most important parameters of the electromagnetic field in terms of bioactivity is polarization in combination with coherence, ELF components, pulsing modulation, the intensity of the field, the exposure duration, and the field variability. Which one of these parameters is responsible for such an intense bioactivity of the mobile phone field? All 6 of them were totally polarized. Therefore, we must exclude polarization as the critical parameter. All of them included ELFs. And 3 of them, they were pulsed on ELFs. And still, the pulsed electric field produced a very small effect. Therefore, we must exclude ELFs as the critical parameter. Mobile telephony fields, they were at environmentally accounted intensities experienced by all users at the very same intensities, because they were emitted by the mobile phone itself, while the other fields were at much stronger intensities than those environmentally accounted. Therefore, I'm afraid we must exclude intensity as well. What remains? Exposure duration. As I told you, with the other fields, with the magnetic field, we had much longer exposure duration, 120 hours. And with the pulsed field, 30 hours, and while with the mobile phone fields, we had only 36 minutes. Therefore, we must exclude exposure duration as well. What remains is field variability. Therefore, the conclusion is that once an electromagnetic field is polarized, includes ELF, and has adequate intensity, the parameter that makes the difference is variability. And going to the explanation of the biological action of the polarized electromagnetic fields, I already told you that all cellular functions are controlled by ion flows, therefore disrupting the ionic concentrations within the cell, we disrupt biological functions. And that's how man-made fields work. They irregularly gate electrosensitive ion channels or voltage-gated channels, and therefore irregularly change the ionic concentrations within the cell. Professor Paul will explain you what can happen after that. This is the structure of a cell membrane with the lipids and the transmembrane proteins. This is the structure of an ion channel consisting of parallel alpha helices and forming a pore in its interior through which the ions pass through. This is when we open an ion channel, which is a protein. There are several groups, not the same in every cation channel, but basically all electrosensitive channels, they have identical, they have very similar operation. They all have some charged helices due to the existence of charged amino acids. These are fixed charges on the helices, which are considered to be the sensors, the electrical sensors of the voltage-gated channels. There were amazing experiments in the 80s and 90s which revealed the structure and the function of the voltage-gated channels. We owe a lot to these experiments. We know so many things. We wouldn't be able to give an explanation if we didn't have this information. And they found that in order for a voltage-gated channel to be gated, we need to have a change in the membrane potential in the order of 30 millivolts. The normal value of membrane potential voltage is 100 millivolts. So this is the 30% change I was telling you before. And they even calculated the effective charge of the sensors, which are called S4 helices. They even calculated the effective charge of the S4 helices to be 1.7 times the elementary charge, the electron charge. So we used all this information to formulate a mechanism. A mechanism in science, in physics, has to be quantitatively expressed, not simply by words, not qualitatively. This is a principle in science. And how can a theory, a mechanism, be expressed quantitatively? This means it has to be expressed by equations, and not just equations. The equations must be solvable. Therefore, by equations and numbers. But in order to solve the equations, we got to think what kind of forces we have, and we have to collect all the forces that we know from oscillation mechanics. The force from the external field, a damping force necessarily, because we have friction everywhere. We have friction, we have damping, we have resistance everywhere, in every movement. And the restoration force. These are the standard forces from oscillation mechanics. But we have some parameters here that we need to calculate. We need to know their values in order to be able to calculate these forces and solve the equations. How much is the value of the damping coefficient? The damping force is equal to damping coefficient times the velocity of the ion. We're talking about ions passing through the channel. And the restoration force, we know from standard oscillation mechanics, depends upon the mass of the ion and upon the cell frequency of the ion. What will be the cell frequency of the system? Every oscillating system in nature has a characteristic cell frequency which is unique. If we take a tree and we hang it from a building and we push it to let it oscillate, it will oscillate with a unique frequency. Another system, we take a stone and we make it oscillate, will oscillate with another frequency, unique. Every oscillating system in nature has a characteristic frequency of oscillation when it is left free to oscillate, which is called self-frequency. So what are the self-frequencies of the ions? Fortunately, thankfully, there were amazing experiments that had found that the ions within the cells do not move randomly, but they oscillate very slowly with frequencies on the order of 0.1 or 0.01 hertz. And each ion, potassium, calcium, magnesium, it's cation because we're talking about voltage-gated channels, mostly cation channels, has its unique self-frequency. This is amazing. But all self-frequencies are close to these values, 0.1 to 0.01 hertz. So we adapted a reasonable value of 0.1 as the self-frequency. And we assumed that the self-frequency of the ion, which is forced to oscillate by the external field, the self-frequency is represented by the spontaneous ionic oscillations. That was a very reasonable assumption. Therefore, we were able to solve the equation, which is, where is the equation? Here is the equation. We have all the forces, and we're able to calculate the different parameters. Previous attempts, there were very important attempts before. They failed because they did not take into account damping forces and restoration forces, and they did not take into account polarization, which creates coordinating forces from several ions simultaneously on the channel sensor, in contrast to the thermal motion, which is much stronger motion than the forced oscillation, but it's random, so plays no biological role. And we had to calculate also the damping coefficient. We had to calculate. First, we had to calculate the maximum ion velocity, and there were other amazing experiments with the so-called patch clamp technique in the 70s. They used micropipettes, tiny pipettes, and they isolated a single ion channel. They depolarized the membrane, and they measured the current, the electric current passing through the open channel. But we know the charge of the ion, so we know how many ions they pass per second, since we have measured the current. And we know that the ions pass one by one through the channel in single file. Therefore, that's how we calculated the velocity of the ions. And in this way, we were able to solve the equation and form the mechanism which finally shows, you can read all this in my publications, I have no time, finally shows that the bioactivity of a field is proportional to its intensity and inversely proportional to its frequency, meaning that lower frequency fields are more bioactive, in agreement with the experimental data I told you before, and that pulsing fields are twice as bioactive as continuous fields, because we have a constant term in the equation that doubles the amplitude each time we have a next pulse. I will pass these slides and I will go to the conclusions, because I have no time. I will go to the suggestions. Avoid any unnecessary exposure to man-made electromagnetic fields. Avoid living close to any antennas or high voltage power lines. Make prudent use of mobile phones. Make use of the loudspeaker or air tube headsets. Keep the handset at least 40 to 50 centimeters from the body when you talk. Minimize number and duration of calls. Do not carry the device on your body while it is on. Put it in airplane mode or turn it off. Keep it at the greatest possible distance during the day and turn it off during sleep. Use wire connections for domestic phones and internet. Disable Wi-Fi and Bluetooth from cars and devices. Even printers, they have Wi-Fi and Bluetooth. Educate parents and pregnant women not to expose embryos and kids. Avoid shielding, that's my suggestion, as this may cause internal desynchronization, because it prevents Schumann oscillations to enter the space, to reach our bodies. And urgent need for drastic reconsideration of current exposure limits, especially for RF and microwave bands. Thank you very much.